digestion in the stomach. We ended up with digestion in the esophagus. What happens in the esophagus? So from the esophagus, food will go downwards to the stomach and into the stomach, which is that sac that we have here. For food to leave from the esophagus and into the stomach, we have a ring of muscles here that we refer to as the cardiac sphincter muscles that form a valve. A valve because these rings can contract and close up that entrance or relax to open up the entrance. We have another ring of muscles here. I won't mention it again, so remember, when food needs to leave the stomach and into the duodenum, the first part of the small intestine, that ring of muscle is called the pyloric sphincter muscle. It will also do the same thing, contract to close up that space or relax to open it up to allow food to leave the stomach and into the small intestine. So at this point, the cardiac sphincter muscle is the one that's opening so that food can leave from the esophagus and into the stomach. That's how food is delivered from esophagus and into the stomach. Now, in the stomach, we have two forms of digestion again, mechanical and chemical. Mechanically or physically, the stomach contracts because the stomach wall is made up of muscles that will contract and move the food, the content or the ingesta around by the action of peristalsis. The churning movements of the stomach wall will then cause the food to break down much further, and this will also cause the food to mix properly with gastric juice to form chyme. Chyme is the mixture of the ingesta with gastric juice, which is then churned into a pulp. So it forms chyme, which is much more liquid than what we had in the mouth. The hormone gastrin is secreted by the stomach walls, and this happens when proteins are detected in the stomach, because inside the stomach, that's why we have digestion of proteins occurring. In the mouth, we were looking at digestion of carbohydrates, but in the stomach, this is where digestion of protein occurs, and digestion of protein had not yet occurred at the level of the mouth. So this is where for the first time it would start generally. So whenever protein is detected, whenever proteins in the food is detected in the stomach walls will produce the hormone gastrin, which will pass through the bloodstream and come to stimulate again the stomach cells, the cells of the lining of the stomach, to then produce gastric juice, which is composed of different forms of liquids that we'll explore in our next slide. Gastric juice contains then 4 components: hydrochloric acid, protease enzymes, mucin, and water. Those are the 4 major components. Hydrochloric acid is secreted so that the environment can be made acidic for the enzymes for protein breakdown. The enzymes that are involved in protein breakdown prefer to work in an acidic medium, contrary to the enzymes that we saw in the mouth salivary amylase that prefer to work in a neutral or alkalinic environment. So since the food coming from the mouth is more or less alkaline, the hydrochloric acid is produced so that that food can be rendered acidic for the proper functioning of protease enzymes, which prefer acidic environments. Secondly, hydrochloric acid acts as an antiseptic because it helps, being an acid, to destroy germs which must have come through along with the food so that they don't become pathogenic in our body, so that they don't cause diseases. The protease enzymes, two major protease enzymes that are found in gastric juice include renin and pepsin. Pepsin is an active form of pepsinogen because pepsinogen is the one that's originally secreted in gastric juice. But due to the action of hydrochloric acid, pepsinogen is converted to pepsin, which is active. Pepsinogen is inactive. It means it cannot break down a protein, but it needs to be converted to pepsin before it becomes active and starts working as a protease enzyme. The reason why pepsinogen is secreted such that it's not yet active is because gastric juice is secreted more or less every time in the stomach. So then, understand again, the stomach itself is a whole pack of proteins. So, if pepsinogen, always present in the stomach, is active, then if there's no source of food containing protein, then the pepsinogen itself will start breaking down the walls of the stomach, which are mostly proteins. That's why it is secreted in an inactive form, so that only when there is food containing proteins will it be activated to form pepsin, which will then act to break down proteins as a protease enzyme. Renin mostly breaks down the protein in milk, which is called casinogen, breaks it down to casein, and casein is then again converted by pepsin to amino acids, because all proteins are broken down to form amino acids, which are the building blocks of proteins. Remember, carbohydrates in the mouth were broken down to glucose, which was the simplest unit of sugar. Mucin, also produced in gastric juice, will then form a thick protective layer on the inside of the stomach, protecting it against enzymes, against protein enzymes, like what I was saying earlier. The wall of the stomach is made up of proteins itself. So mucin must be produced so that the walls can be protected. It's because of the effect of this gastric juice on the walls of the stomach, the enzymes and the acid itself, that we have that sense, that burning sensation in the chest that we usually refer to as heartburn, although it's not the heart that is burning. Additionally, water is added to the food to make it fluid and easy to move within the alimentary tract. When all these have been done, the action of enzymes and the mechanical actions have been done such that the food is churned enough and fine enough and well mixed with gastric juice, the pyloric sphincter valve, the other ring of muscle that I mentioned earlier, will then open up so that the content of the stomach, the food, can be emptied into the duodenum, which is the first part of the small intestine, and this is done by peristaltic movements. In the small intestine, the main function of the small intestine is rather the absorption of nutrients that have been broken down into their basic components. Absorption of the nutrients is then assisted by mechanical contractions of the intestinal walls. That's still peristalsis. That's happening all along the digestive tract. Peristalsis ensures that food is thoroughly mixed with pancreatic juice, intestinal juice, and bile. It means it includes peristalsis in the stomach as well. In this case, now in the intestine, we're looking at intestinal juice and bile. The nutrients are then absorbed by the small finger-like structures that are present in the walls of the small intestine. The mucosa, if you remember, the inner wall. These structures are called the villi, singular villus. And the nutrients are absorbed by three processes, either diffusion, osmosis, or active transport. You should have done diffusion in grade 10. Otherwise, diffusion is the movement of molecules from their regions of high concentrations or from areas of their high concentrations to areas of their low concentrations along a concentration gradient until equilibrium is reached. It means, for example, if there is a lot of molecules, if I have two sites, A and B, and I have 1, 2, 3 molecules and 4, and only one on side B here, if diffusion has to occur, molecules will not move from B to A. No, molecules would rather move from A to B. The reason is because A has many more molecules than B. A has 4, B has only 1. So how many will be moved? Let's say A has 5. How many will be moved from A to B? We'll move going to move 1. Remember, A had 5 and 2. We're going to move only 2. Why? So that B can have the 1 and 2. Why? Because we want to reach equilibrium. Equilibrium means balance. Both sides now have equal numbers of molecules. 3 this on A and 3 inside B. And like I said, this movement is done along a concentration gradient. It means it moves from where it was too much at A to where it was less. And diffusion is kind of passive transport. Passive because it doesn't use energy. You don't need energy to move molecules from where there are too many to where there are few, because when molecules are crowded in one place, they have that natural tendency of wanting to move on their own to where there is less population. So that's what happens. In this scenario here, now this is 1 and this is 2. In my scenario 2, I have now more molecules on side B than A. So what will happen? One molecule, only one. This time molecules will not move from A to B anymore. No, because B has more this time. So one molecule from B will move to A until equilibrium is reached. So now we'll have 2 and 2. If I have scenario 3, I have 1, 2, 3 molecules here, and 1, 2, 3 molecules here. The question is, will there be diffusion? Answer is no, because there are 3 on side A and 3 on side B. There's already equilibrium. That is the way food diffuses from the intestinal lumen and into the walls of the villus. While food is moving, this could have been, let's say, side B here that we had before. At A we had 5. So let's say this was A here represented, number 1 here represented glucose. It means within the walls, within the capillaries of the villus, at B here there was less glucose, but in the lumen of the small intestine there was more glucose, up to 5 molecules. It means glucose will move from A to B. In other words, glucose will diffuse from the walls, from the lumen of the small intestine to the blood vessels in the capillaries. That is diffusion. That's how nutrients are taken in to the villus. Maybe at 2 here, where we had just 1 this side and 3 the other side, maybe there was too much waste in the blood vessels. Waste products such as nitrogenous waste or urea and less waste here. If this was the excretory system, for example, then the nitrogenous waste will move from side B to side A so that it can be excreted. This is done by diffusion. Now, osmosis. Osmosis is exactly the same way as with diffusion. It is also passive. But the difference here is just that diffusion could be molecules like nutrients or gases or liquids. But osmosis is essentially dealing only with water. That's why when we see the prefix osmo, osmo is like attached to water. So all what will be happening with the movement of water molecule, will term that osmosis instead of calling it diffusion. So osmosis is the movement of water molecules, not just molecules as we said with diffusion, the movement of water molecules from their regions of high concentration to their regions of low concentration. Active transport is the opposite of diffusion. Active transport. Active means it needs energy. It is not passive like with diffusion. It needs energy to transport the molecules, even against a concentration gradient. With diffusion, molecules are moving along a concentration gradient, but with active transport, it is a movement of molecules from their regions of low concentration to regions of high concentration where they're already too much, against a concentration gradient. Because when you move along a concentration gradient, you're living from high to low. But when you move against a concentration gradient, you're moving from low to high, and in that case, you have to force these molecules to move. You have to force them, and while you're forcing them, it means you will have to use energy because you're using force. Now, if I go back to my scenario A here, now I have 4 molecules here. 1, 2, 3, and 4. And at B, I have just 2. But say the body is craving for something and it will not let it go. For example, vitamins. Even if it has already some of it, it will still want more. But this time, because the two sides are not balanced, where it wants to take the molecules to side B, there's already some there. Let's say at B, there is 5. And at A, let's say at B, there's 6. And at A, there's just 4. At side B here, there's already too much. But yet, we want more vitamins from side A here. It means we'll have to forcefully bring these molecules. And this can only happen through the use of energy through active transport. So B already had 6. We're going to take more or less everything from A to B so that we now have 10. This will only happen with use of energy, active transport. So these are the 3 methods by which food, nutrients will be absorbed in the small intestine. Chemical digestion in the small intestine. When food is detected in the small intestine, the walls of the duodenum, which is the first part, remember the first part of the small intestine here, the walls of the duodenum will then produce or secrete the hormone secretin. And the hormone secretin will travel through blood vessels and go into the pancreas. When the hormone secretin goes into the pancreas, it will then stimulate the cells of the pancreas to produce pancreatic juice. Then pancreatic juice will then move from the pancreas through the pancreatic duct, passing through the pancreatic duct, where it will meet with bile coming from the common bile duct, and the meeting point will be at the hepatopancreatic duct, and all of that pancreatic juice and bile will then be delivered to the duodenum.